Hey, welcome back to the second part of our conversations on changes that occur with chronic pain. And so today, or this module, we're going to be chatting about changes that occur centrally um, in people with chronic pain. And by centrally, we're going to be talking more about changes that occur in the cortical and subcortical regions. Even though theoretically we did talk about some changes that occurred centrally in the spinal cord in our last module, this module is going to focus primarily on the changes that occur in the cortical and subcortical regions. And this is where a lot of the science is changing, and a lot of the growth in terms of our field of pain sciences are really starting to shift and inform the way we practice. And so exciting to start to share this information with you and realize that this information is changing at an astronomical speed. If I look at research on almost a monthly basis, each new journal periodical helps inform us of some of the amazing changes that are occurring in the brain and in the subcortical regions. And a lot of this information and these changes have occurred because the technology is finally there for us to start to take a look at some of the changes that are occurring in these regions. And so over the next few minutes, we're going to be talking about some of these changes. So if you remember some of the talks that we've had in the past about the history of pain sciences, if you remember Ronald Melzack, he proposed a kind of a theory to explain some of the changes that occur not only in the periphery but in the brain as well, and how he believed that all of them were kind of linked together in this neural matrix of structures. Sometimes this neural matrix is called a neurosignature. What we're really talking about is this pattern of structures that are recruited in people with pain. We talked in the past about this neural matrix as it relates to nociceptive pain. And now we're going to take a look at some of the changes that potentially occur in people with chronic pain, because there are some distinct changes that occur in people with chronic pain. Remember in the past too that we've had other scientists come along and rename these neurosignatures and with other nomenclature. And so both Lorimer Moseley and David Butler coined the term neurotag, very similar to the neuromatrix, very similar to the neurosignature, just different name for the structures that are recruited within the brain in response to nociception. So what are some of the changes that occur in the cortical and subcortical regions? Remember, if you look back at our overriding concept of how things work, we're really looking at the perception phase of things. So not looking at the transduction, we're not looking at the conduction, we're not looking at the transmission types of structures. Instead, we're looking at the science of perception, or those changes that occur within the cortical and subcortical regions. Cortical regions function more analytical thought and more conscious levels of thinking and reasoning and abstract thought, whereas the subcortical region, if you look at the picture on the right, is the regions that are deeper. A lot of times it's the more primitive regions of the brain, and these subcortical regions of the brain are designed more for unconscious thought. So these are the things that occur more at an automatic level. Things like heart rate, things like respiratory rate, things like blood pressure. These are all regulated, but they're regulated below our level of consciousness at the subcortical regions. So what kind of changes occur in the cortical and subcortical regions? Well, changes occur really at two levels. They occur at the level of activity, and changes occur at a structural level as well. So you'll see changes, and we'll discuss changes that occur in both of these types of structures, or not structures, but changes that are occurring within the brain. So in our prior lecture on acute nociceptive mechanisms, we talked about the different systems that are potentially recruited within the brain. We talked about how there's a primary, secondary, and tertiary system of brain activation that occurs in people with in response to nociception. Those nomenclatures were based primarily on physiological mechanisms or neurophysiological mechanisms, and were related more in terms of the speed in which these areas were recruited. If we take a step back and look more at the function of how each of these systems are recruited, we get more of a meaningful pattern that we can use as clinicians as we start to take a look at our patients and to help us explain what is happening in people with pain, as well acute nociception as well as chronic pain. So if we look at those systems in another light, we can look at them as three modes of network brain activity. There's a default mode, there's a saliency mode, and there's an executive mode of activity. And each one of these have distinctly different responsibilities and functions. If you look at the default mode really refers to that mode of activity that the brain is working automatically. If you think about it, our brain is never silent. There's always levels of activity occurring within the brain, and a lot of it occurs at the subconscious level, where the activity, you're hearing things, you're smelling things, you're seeing things, and the body's responding without you having to think about it. Even at rest, when you're sleeping, the brain is still taking in information from its surrounding and processing it. This baseline level of activity required for us to survive is called the default mode network. Sometimes this default mode is just when our mind is daydreaming or wandering, or we're not really thinking about anything. The default mode is the area that's activated when this occurs. Now, the salience mode is kind of an intermediate mode between the default mode and the executive mode. You know, our brain, our senses, all of our resources are always scanning the environment to look for a threat and look for changes in the environment. And what happens is the brain takes in this information from the periphery. If it's something that is not really that important, then what will happen is we'll stay in the default mode. But as the saliency network is activated and it's continuing monitoring, if things become out of the ordinary, if things become unique, then what happens is it starts to turn on systems and it starts to activate other systems. So if you start to take a look at the saliency network is really kind of like our body's early warning sign. It's the thing that's monitoring our environment to see what's going on and looking for change. The final network is our executive network. The executive network, what its job is to, if there is change that's occurring in our network that's picked up by the saliency network will activate the central executive network. And the central executive network are those areas that we're more accustomed to thinking about for conscious thought. These are our higher levels of processing. And so what will happen is the central executive network gets activated and the brain decides what its appropriate response. A lot of times the saliency network will not engage the central executive network for a lot of things that aren't that important, because what it'll do is simply activate some of the lesser systems, and no conscious thought is necessary. So those are the three main networks: the default mode network, the saliency network, and the central executive network. So once again, the default mode network, monitoring, maintaining base level of function. The saliency network, scanning and monitoring the internal and external environments, and functioning as a warning system. The structures involved are usually the amygdala, the anterior cingulate cortex, and the insular cortex. We saw activation of all that occurring with the nociception. And the executive network of the brain, This supports sustained focus and working memory. It engages in tasks that require rational thought and conscious control. So those tasks that are in response to things that are unique or require our brain to have further action taken, this involves the executive network. And sometimes it involves suppression of impulses as well. You'll see on the picture right, these require those parietal and prefrontal areas of the cortex that we see activated in our patients with nociception. So just a summary slide that tells the function of these different brain networks. The default mode, the saliency mode, and the executive mode, and the various areas that are kicked in. The default mode maintains homeostasis. The saliency mode monitors the environment, involves the anterior cingulate cortex and solar cortex and anterior cingulate cortex. And then the executive network responds to these changes and unique instances. And once again, the medial prefrontal cortex and the parietal cortex. So what happens in people with chronic pain? Well, it's very, very interesting. As we start to take a look at MRI studies, we've found a common pattern in people who have chronic pain. Now, the areas are not activated identical in any two people. And that should make sense because no two people have the same type of experiences. No two people have the same type of input. No two people have the same type of circumstances and situations happening to them at any one time. So it makes sense that no two people have exactly the same recruitment pattern. And it can be argued that even in a person, very rarely do you have the same exact activation pattern because in people, circumstances change from day to day and second to second. But what we do find is if we start to take a look at large samplings of MRI in people with chronic pain, we start to see certain patterns occur in our patients with chronic pain. And so if you look at the illustration on the top, actually what you see is some changes that occur in healthy brains as a result of nociception. So if you put them in a fMRI machine and you get a little probe, you give them pain in their finger or their foot, and you keep pushing with the pressure until they say, ow, that hurts. You'll see this kind of activation pattern. And this is our normal activation pattern that we talked in prior studies or prior lectures, where you take a look at some areas light up, like you can see the anterior cingulate cortex, the insular cortex light up, and you'll see other areas stay cool and calm. Look at that in comparison to the changes that you see or the activation you see in people with chronic pain. In people with chronic pain, you do the same thing. You stimulate them in their foot or hand. You push on it until they say that hurts. You'll see how the brain is almost amped up, how there's less level of decreased activity and huge amounts of areas with amped up activity. And a lot of the change occurs in the saliency network. So our brain is overstimulated, it's always active, and you can see that kind of in our patients because what's happening is that the area that's job is to look for a threat, to scan our environment, to see if anything is novel is occurring, is acting, has a tremendous amount of activity compared to the healthy people. And that you can see that in our patients where they're always scanning the environment. Their system is always hyper-tuned and hyper-alert and hyper-aware. It's like having an alarm system that is too sensitive or an early warning system that is overactive or too hyped up. So that's what happens in people with chronic pain. You could also see how the prefrontal cortex is always having to make more and more executive decisions based off the information being routed to it from the saliency network. And so in people with amped up brains, you'll see an increased activity. Really, one of the interesting things, too, is not only is there increased activity, but there's an increased duration of activity as well. These areas light up, and in healthy people, as soon as you remove the stimulus, these areas will return to a normal default mode. But in people with chronic pain, you give them the stimulus, And what happens is their brain overacts and it stays active for longer and longer periods of time. So it has difficulty returning to the restful state, the default mode. We also said that in beyond levels of activity, there's changes that occur in the structure of the brain as well. And we'll find that in a majority of structures of the brain, those areas that are most engaged in nociceptive processing become atrophied in people with chronic pain. And so there's decreased gray matter in the medial prefrontal cortex, the thalamus, the anterior cingulate cortex, and the hippocampus. The one structure that we actually find hypertrophy is the amygdala, and we'll talk about that in a second. So a majority of the original research that was done on these changes that occur in people with chronic pain was actually done on a structure called the hippocampus. Now, the hippocampus is called the hippocampus because it's shaped like a seahorse. And what it does is it wraps around the amygdala. And if you remember the amygdala, which we'll talk about in a second, the amygdala is really a structure designed for dealing with emotions and threat. So sometimes it's been associated with the emotion of fear, but it's not a structure that's solely responsible for fear. Fear is one of the things it does, but there's also other intense emotions that are related to activity in the amygdala as well. So really, if you think about the amygdala, its job is to respond to threat. And so the amygdala, the hippocampus, really wraps around the amygdala, and its job is to act as kind of a short-term to long-term storage facility for the activity that's occurring in the amygdala. It'll also store information from other parts of the brain, but one of the things it does is it stores some short-term memory about fear and threat from the amygdala. And it's also critical for temporal memory. And so the amygdala or the hippocampus' job is to give us contextual relevance to the information coming in. So if we're threatened, if we have fear, what its job is to give it a time and a place and a context. That is the job of the hippocampus, is to store those memories. We find in patients with chronic pain that these structures are actually atrophied. They're actually smaller. And the very interesting thing is that decrease in size is actually, we can find that long-term after we've had a threat or injury. It's interesting if we take a look at children who've been traumatized when they're young. We'll find that if we study their hippocampus as they get older, it is smaller, especially in people with post-traumatic stress disorders from a younger age, and actually in people with chronic pain, we see a similar pattern of atrophy within the hippocampus. The one structure that we see larger amounts of growth in the structures is actually the amygdala. And remember, I just talked about how the amygdala is responsible for the first processing of information about threat and fear. And so it processes these events, and then it sends it for storage, and it sends it out to other areas for appropriate responses. In this area, it's actually hypertrophied in people with chronic pain. So the question is, why is the amygdala hypertrophied and all other structures atrophied? Which doesn't make sense if you look at it from a structural perspective, where you would think that more activity in an area would actually result in a hypertrophy in an area. But if you think about it, that is really true of orthopedic and musculoskeletal problems, right? If you work a muscle more, if you work a muscle more, it hypertrophies. And so that's where you get the common concept that structures that are used more get larger. You have to understand that it's a little different when you start to look at the structures that happen in the brain. brain's structures are really based off neurons, but also about the myelin sheath that surrounds the neurons. And so as structures are utilized more, part of the learning process is to increase the amount of myelin that surrounds a neuron. It promotes faster use of a certain neural pathway, and so it increases the size of the myelin sheath around a neuron when it's activated more. And you'll say, well, that should mean that neurons are activated more, so it should increase, just like muscles. And that is true. But what happens the theory is, what happens in people with chronic pain is that, say, an area like the hippocampus or an area like the insular cortex, generally in healthy people, what happens is new pathways are being formed all over the place to any specific stimulus that comes in. And so all areas of the hippocampus or all areas of the insula are being recruited and staying healthy. What happens in people with chronic pain, we're thinking, is they use the same pathway over and over and over again, and they don't deviate from that pathway. And soon what happens in that pathway is that one bad pathway continues to get utilized more and more, whereas the pathways that are on either side of them and surrounding don't get activated. So those areas start to atrophy from disuse. So why would the amygdala get bigger? Well, we're thinking that the amygdala gets bigger because this is our threat and fear processing center. So what happens is everything gets routed through that because our saliency network is overactive and sending everything as a potential threat to the amygdala. And so that's getting overactivated. Once again, this is purely a theoretical activity at this point, but it does make some sense in terms of what we see from a functional standpoint. We used to believe that pain is in the brain, which really we'll see in future research or future modules that we start to question this whole theory that pain is in the brain. We agree that pain is processed in the brain, but we really think that when you start to take a look at that platitude may not necessarily be accurate. Because if this were true, then the bigger size of the brain, potentially the bigger amounts of pain something would have. So things like elephants and would have more pain, whereas animals like chimps and cats would have less pain because of the number of neurons. So, but it's not the size, it's more the amount of neurons that are potentially processed. Humans have the greatest percentage of neurons, and that's what the majority of those neurons help us to process information and to be able to think with our executive functions and make logical decisions that other animals can't necessarily make. And so it's not necessarily the size of a brain that's important, it's more the amount of neurons. So we just talked about hypertrophy and atrophy. So we kind of proceeded to go a little quicker than that. But one of the things that, in terms of the mechanisms involved in hypertrophy and atrophy, we talked about the increase in myelin size. And once again, that goes to glial cells. So if we look at glial cell activity, remember I said we were going to be talking about it in this module. Really, we talked in the past about microglia and its impact on the peripheral nerve. But also you can see how there's oligodendrocytes. We talked about that, and they're responsible for the myelin through the Schwann cells. And then there's also ependymal cells, and they're responsible for cerebrospinal fluid. And then we have astrocytes and satellite cells, and their job is to help repair our nerves. Now, what happens in our brains that cause the atrophy and causes the hypertrophy? We're not quite sure at this point, but it's interesting lines of research are taking a look at the role of glial cells in this component. Because if you think about it, only about 15% of our brain is actually nerves. We have a tendency to think our brain is 100% nerves. But if you take a look at our brains, only about 15% of it is nerves themselves. A majority of the remainder of it is actually glial cells. And so glia actually means glue. And so it's the thing that binds our neurons together. We used to think that they were just purely support cells, but now we're realizing they have a function in communication as well. And we're thinking that the health and the development of things like the Schwann cells around our neurons are an important part of the aspect of chronic pain, because what ends up happening is these cells, they're responsible for a lot of learning. Like the Schwann cells, what they'll do is they'll maintain a healthy nerve around that pathway that's being recruited over and over and over again. But in other areas, they'll get smaller. This is important because the increasing of size and health of a Schwann cell is important for learning. We talked about how this process is hugely important in helping facilitate activity in a connected network of information, and that's what learning is. It's taking information from a whole bunch of areas, it's taking it and putting it into our neural pathways, and then storing it into memory, and it's facilitated when it's activated more and more. That's the process of learning. And as it's recruited more and more, then what happens is that system becomes healthier and healthier, and most of that work is done through the Schwann cells, and as well as the microglia and astrocytes helping maintain the different neurons. We'll actually find some very interesting things that occur actually in other aspects of the brain too. There's changes in laterality. So generally the brain communicates between both sides of the brain pretty actively when it's making decisions. We find in people with chronic pain, they'll stay in one side of the brain more, whether it be the right side or the left side. People sometimes will stay in their more logical side. That's the left brain, which is responsible for logic and speech and analysis. So sometimes your patients, they'll be living in that side, and you can really see that in your patients as you talk to them. And other people will stay primarily in the right side of the brain, which is more creative, intuition, survival-based, art-based. And so you'll see people activated in chronic pain and staying in that side and not crossing between the two sides of the brain. And so you'll see that pattern in people, and you'll see changes in atrophy and hypertrophy that are specific to laterality as well. There's also something that we find, Laura Mosley's team and David Butler's team have coined the term cortical smudging, but really what it is it's areas of the brain that become less specific. Generally, if a sensation is sent up to the brain, you'll see it activates a very specific part of the homunculus. You'll find in people with chronic pain that area of activation becomes wider and broader. And so no longer is it a crisp activation. It's spread out over a wider period of time. And so that accounts for some of the things we see in patients where they're not able to accurately describe an area of pain. They'll say it's in more general terms. Instead of a finger being sore, they'll see a whole hand feels sore. And so that's some of the clinical manifestations and how that is actually being seen from a neurophysiological standpoint in the brain, because it's no longer this crisp defined area of the brain. It's more smudged. And you'll see that relevant, or you see that play out in the fact that they cannot two-point discriminate very well. What you'll see is you'll put pressure on 2 points of someone's finger that's healthy, and they'll say, oh, that's 2 points. And people with chronic pain, they'll say, I just feel 1 point of activity or 1 point of pressure. And so that's a common test that we'll run to see if there's been changes that occur in terms of the brain's ability to differentiate. And sometimes they'll use that as a clinical tool to help people start to regenerate that ability to feel 2 spaces again. And it's actually interesting that people will have difficulty visualizing themselves as well. So if you ask people to draw pictures of themselves, a lot of times they will draw themselves fairly nicely as a self-portrait, but they'll have difficulty drawing the area that's damaged. And we believe that's because that they cannot visualize that area that well. It's become smudged and difficulty to differentiate those areas in the brain, and they really don't have a picture of what that area of the body looks like anymore. And so part of our job is to help them remember what that area of the brain looks like. And so there's different types of treatments that we'll use to help us help a patient learn to visualize that area again. So as we start to wrap up this module on changes that occur in the brain and the subcortical regions, realize that there's a huge amount of information and changes that are coming across yearly and changing the way that we see chronic pain and some of the changes that recur in the brain. This is the one big area of research and exciting changes that occur because technology has given us an insight and a look into the changes that occur in the brain. Now, as we start to take a look at these changes, it's important to realize that these are just changes that are occurring from a structural standpoint. They're changes that occur neurophysiologically in the periphery as well as the brain. Remember that these changes are occurring with signaling. The brain changes are changes that are occurring with perception. But still, at this point, they don't necessarily mean that someone's going to have pain. All they do is an indication that there's changes that are occurring neurophysiologically. Really, the changes that occur with pain has to do with how the information is processed and the interpretations that's being made with those signals. And that goes back to the purpose of pain that we've talked in past modules about. So as we start to take a look at the wrapping up this module, just a chance to reflect back on some of the information that we've gotten in the past about the purpose of pain. Remember, overridingly, the purpose of pain is to initially protect and survive. And as our body learns, as our brain learns, then what happens is afterwards, it's designed to warn and prepare. And so a lot of these changes that you'll see happening neurophysiologically will be actually evidence of learned responses, neurophysiological responses that are designed to help with this secondary warning and protection mechanism, preparedness protection mechanism. So that's the end of this module on changes that occur in the cortical and subcortical regions, and the previous module that looked at changes that occur in the periphery. We'll finish up this module with those talks, and thanks for joining in, and we'll see you all soon.